So good morning, everybody. My name is Marina Lipizer. I'm from the Italian National Oceanographic and Geophysics Institute, based in the north of Italy, in Trieste. And my task within EMODnet Biology is to lead the work package dedicated to uptake and outreach. So to facilitate the connection between EMODnet data products, and so the scientific parts with the end users, with with the European directives, but also with the scientific community, to facilitate data sharing, but also data use and reuse also, which is the philosophy of EMODnet. I would quickly go through the agenda of these 2 days, 2 mornings, starting with how we arrived here. Within EMODnet, we dedicated a time to map our stakeholder community. So who are the institutions who can possibly obtain benefit from participating to EMODnet? We have prepared some questionnaire in order to try to understand what the needs of our stakeholder are. And many of you have participated to our questionnaires. And so we thank you for your participation, because these feedbacks are very useful to tune our current and our future activities. Then we organized a first workshop, which was specifically dedicated to the regional sea conventions. It took place in Costanza and online, and it took place in April this year. And sorry, I'm letting people participate. And now this workshop, which is dedicated in particular to European projects. As you well know, EMODnet is being proposed as one of the main data infrastructures in Europe. And so connecting data with EMODnet is really considered an important issue. But I don't want to steal too much time to my colleagues. So this is the agenda and how the meeting will be organized. We will have an introduction by our European representatives from DG MARE. And then we will have our coordinator, Joana Beja from Blitz, from the Flanders Marine Institute based in Belgium, who will introduce us EMODnet Biology. And then we have an analysis of the results of the questionnaire, which many of you contributed to. And then we have time for questions. And then we have some project pitches. We have received a lot of availability to present several projects, and unfortunately for time constraints, we had to make a selection. But we think that there will be time to interact and to provide feedbacks also to all the other participants. And then we will start after the short break. We will start with the guidelines, with the tools, with the thing that EMODnet can, the support that EMODnet can provide to projects providing data. So we will have EMODnet Biology data guidance. Then we have some other project pitches. And then we will have a presentation of seabed habitats, which is also strongly connected with EMODnet. And in the next day, we will have another session, also starting with project pitches. And then we will have presentations on what the data resources are in EMODnet Biology, what are the guidance to prepare products, and what are the tools and services which EMODnet has developed over the years in order to support harmonized and standardized data management and data accessibility. So I welcome the many new participants that have been connecting so far. And as I mentioned before, we are recording this meeting in order to keep this information also for the future. And I will now leave the floor to Mrs. Zoe Konstantinou from DG MARE to welcome the meeting. I will stop my sharing, and
thank you, Marina.
Thank you very much, Zoe. Thank you.
I'm here representing DG MARE. DG MARE is the owner and manager of the European Marine Observation and Data Network, but I'm also representing my colleagues in research and innovation, my colleague Nikola from the side of the development and management of research projects. And this is a common message from the Commission. So, as you know better than me, marine data is gold for European marine policy, for the blue economy, for any developments that are related with climate change adaptation, biodiversity loss, and resilience. In this category of data, biodiversity data, marine biodiversity data deserves a special mention because it is complicated to collect, to observe, to manage, to bring together, to standardize. It's very expensive observations, and I'm not going to say even more valuable, but it's more challenging to put together and make available. From the point of view of the European Commission, we try in the best of our ability to fund research that is useful in this sense. And at the same time, we believe that this research should be of added value for other researchers and society in general. And the collected data is a part of this. All European projects under Horizon Europe have the responsibility to have a data management plan. And this data management plan aims to make data available. Now, to make marine data available, the European Union, through the European Commission, has created EMODnet. And EMODnet is an exemplary service that provides marine in situ data, and managed in the last 15 years to make a very big progress in the sharing of this data, to standardize this data, to harmonize them, and make them openly available in the 7th thematic of EMODnet, including biology and seabed habitats that are more of relevance for your workshop today. Our wish from the point of view of the European Commission is that all in situ data that is collected in Europe with public resources should be made openly available. And this is a wish that is being established also through a number of legislation. So we have the inspired legislation for geospatial data, and we also have the Open Data Directive for public data that should be standardized and that should be openly available through appropriate APIs. EMODnet is doing a lot of this work. Is doing it in a network basis, and it's not an established framework for reporting, but nevertheless is doing a lot of this work, and it's providing the opportunity to everybody that is collecting data to make this data available. We have realized that there is a desire also from the point of view of European projects to be more aware of these capabilities and these capacities, and to be introduced better into the methodologies to make this data available through EMODnet, and to develop data management plans. A series of workshops, one of them is this one today, aims to cater to this. EMODnet doesn't have the resources to cater to the needs of each project separately. The European projects need to develop these data management plans and need to take care of the standardization of their data. But in the essence, this is it. This is the basic work. It's not an easy work to standardize data, to change maybe the protocols that you have been using for years in order to follow common standards. Yet it's unnecessary work that you will have to do the one way or the other, and that it gives more added value to the data that you're collecting for yourself and for others. EMODnet data are going to be directly available through the European Digital Twin of the Ocean, and this is another value that we aim to create through the structures that we're funding in the European Union, aiming to provide legacy to the research and the work that you're doing in the different projects. I thank you very much for being here today. I strongly believe that with the lack of resources that we have, EMODnet can support you, the different European projects, in helping you to structure your data management plans and providing you advice, actually. Not doing the work for you, unfortunately, maybe in another life. But I am convinced that working together in order to make more marine data, and particularly biology and biodiversity data available, it's going to be of added value for all of us, for future work, for future endeavors and future projects, and will create a depth in marine knowledge and European seas that is invaluable for conservation, for development, for policy, for anything that we want to do in the future. Thank you very much, and I wish you great success in the workshop.
Thank you very much, Zoe. This was very important to introduce the topic. Before leaving the floor to Joana, I just forgot to mention that we will collect a short information from all of the participants through a Mentimer, but which will be launched afterwards. So now, please, Joana.
Hello. Sorry about that. I just received an email from a participant that didn't receive a link to Zoom. Marina, when I start, I'll send you a link.
Shall I invite somebody?
Yeah, I'll send you the email before I just start, and then you can.
Okay.
Sorry about this. Is very dynamic, as you can see. Okay, there you go. Good morning, everyone. My name is Joana Beja. I'm the EMODnet Biology Coordinator working at LIS. I know some of you, even though I can't see all of you at the moment, but I do know some of you. Welcome to our workshop, and I hope that it aligns with your expectations to some extent. One thing that I would like to reinforce is that we have scheduled several Q&A slots. I would urge you to engage with us. We won't be able to, as you can imagine, answer specific questions if you have them about what you can do with this data in terms of the data formatting or your products, for example. But more generic questions, I think we will have time for those. And if there are anything more specific that we cannot address during the workshop, then we can try and follow up via email afterwards. I think that might be the best option. But I would, I would encourage all of you to indeed ask questions so that you can make the most of the workshop today. So I'm gonna share, I think it's this one screen. And I know that some of you will know this already, maybe all of it, maybe part of it. So I do apologize for that. It's a bit of a repeated information for some of you, but you know, it's for the benefit of all. So wait a minute, because I need to have this. Okay, so start very generically. What is EMODnet? We are an EU service for in situ marine data at the same level that Copernicus is the EU service for remote marine data. Okay, so these are the two EU services for marine data. We are divided, let's put it like that, in 7 thematic domains. So we are, this workshop is organized by the EMODnet Biology thematic lot, and we will have the contribution of the seabed habitats because we considered that seabed habitats data are biodiversity data. EMODnet as a whole is quite a big consortium or network. It has more than 120 organizations involved across Europe. And the main objective is to provide free and unrestricted access to data and data products. We adhere with several standards and data principles. You will hear a lot about this, mostly from Ruben. So I'm not going to spend too much time explaining this, but you will hear a lot about FAIR. You might hear as well about INSPIRE and OGC standards for the products and the machine-to-machine communication as well. So what do we do? So this is a diagram that kind of shows our network, at least the closest network, or just a small scale, because we all connect to many more things. So in blue, you can see all the organizations that are part of our consortium. In pink, it's the EMODnet thematic lots, and some of us do participate in various thematic lots, not just biology. And then you have in goldish yellow, the regional sea conventions, and in black, some of the main, but not the only, but some of the main international initiatives that we also are connected to. And that could be via data exchange or just being present and actively participating in those initiatives. So we have, one of our main objectives is to publish marine biodiversity data and to create biodiversity data products. That is our main remit, which is defined by DG MARE. Alongside this, we do a number of other things, as you will see. We provide training and we create training resources and guidance for both the data providers and the product creators. So I think a number of you will fit in either both of these roles, let's put it like that, or just one of them. So we do have a lot of guidance that I would urge you to explore before you ask questions for support, because in most cases, the answers can be found in the guidance. We also publish a number of informative material in terms of in the outreach that we do. And one of the main remits as well is to support the EU member states and regional sea conventions and EU institutions with their work as well in terms of data publishing. And this is an ongoing work that we do during, the funding period that we have. Just a brief overview on the data publication. We focus on European seas, but we're not limited to these. We do, if you look at the data that we have available through EMODnet Biology, you will see that we have data from all of the world's oceans. But indeed, our remit is the various European seas. And the target in terms of reporting is not just what we focus on as well, but it's 9 sub-themes. So from reptiles to seabirds to fish, phytoplankton, and so on. So those are the main data that
nursery area for each one of the species. So
can you?
I don't see the problem.
Oh, okay. But if you don't, if you have data or products that don't exactly fit these 9 sub-themes, that's not an impediment to have those data published in EMODnet Biology. In terms of types of data, we obviously publish data that were collected through, observations, but also the taxa that is identified via acoustic means, taxa that are tracked via tags, for example, taxa that are identified by imaging methods, and we are hoping that we can publish some genomics data or taxa identified by genomics processes this year, if not very early next year. At the moment, we have, and this is all public information, you can find it as well. We have almost 1,500 data sets that have a link to the data file. So they are ready for download in Darwin Core. And we have almost 50 million occurrence records. If you go to the EMODnet GeoViewer, so the link is there. So this is the EMODnet GeoViewer. You go to the EMODnet Biology theme, and the sub-theme, the layer is called Biodiversity Records. You click on it, you can select, apply various filters and select the data you want to download and proceed with the downloads. So those are the filters as I've shown. You can in the EMODnet viewer. We will just add a bit of a parallel information, is that we will be publishing a report of this workshop, and all these slides will be available. These contain links, so we will endeavor to make these links also clickable through the report, so that you can just follow up and don't have to search too much in the EMODnet website for this information. These two slides just show the data increase or the data holdings per sub-theme and per region, depending on which one you're looking for each phase. So typically a phase for EMODnet is funding for 2 years, which can be extended for another 2 years. And we are currently in phase 5. We will end this phase in May next year. And the different colors correspond to the data increase per phase. As you can see, we have a considerable amount of data for benthos and birds, for example, but not so much as for reptiles, which can be understandable, I guess. But we're also missing a lot of data for mammals. And we don't have a lot for angiosperms. And in terms of the regions that we're lacking data, it's the Black Sea, Mediterranean, and the Arctic. These are really areas where we don't have a lot of data. So if your project is working in these areas, we are very much interested in any data that you might have, and we'll be sharing. A brief overview of the products that we have, and do note that most of the products were created by our consortium throughout the phases that we have had in EMODnet. So we have at the moment, and this is a number that continues to grow through time, but we have 38 products, and 3 of them are from external sources. So other projects or other initiatives that we consider have relevant products that we can republish through EMODnet Biology. And in some cases, they are available for a query subsetting through the EMODnet viewer as well. I'm not going to spend too much time with this, just to say, because one of our colleagues, Salvador, is going to explain a bit more in detail about the products tomorrow, is that we also produce guidance in how to create the products so that they can be published through the EMODnet portal and various systems. This is a guidance that our consortium partners need to adhere to. If you don't have such guidance within your projects, I would urge you to also check what we have available and adjust to your needs and to the projects, because that really does facilitate the querying via either the machines or manual via an interface like a viewer like this. Data flows, very generically and very, very high level overview. So we have EMODnet Biology. We receive data from either our consortium partners or external providers, and external providers can be anything from other EU-funded projects to citizen science activities to NGOs that have collected data and want to share their data to other organizations, or so it's quite a wide range of external providers. So we then publish the data in EMODnet Biology, and that data are made available to, or shared, let's put it like that, with OBIS and GBIF, which I hope all of you will know. And it's also available through the in the EU DTO through the Edito Infra project. So basically what this means is that by submitting data to EMODnet Biology, you do it once and the data are shared through multiple infrastructure via machine communications and machine protocols. So you only have to do it once for it to actually be much more widespread than just EMODnet Biology. So the first slide, there's only 2 more slides if I'm not mistaken, and this is what we will not do for you. Following our survey asking a number of questions for feedback, we understood that there is a bit of a expectation on what we can do, on what we should do. So I want to make it quite clear and follow up on Zoe's words that our resources are quite limited in what we can do for the EU-funded projects. We cannot follow up on your data management plans. That is something that you have to organize within your own projects. And we will not follow up on your data. It's quite a time-consuming exercise to do that, and it's really not within our remit to do so. That is something that you will have to do within your projects and discuss with your project officer and your PIs if you are present, and your data managers. We cannot do the reformatting of the data, and I put just data here, but that will be the same as for the products, as you see in the last line. That is not work that we will do for you. If you are submitting data, or your data will be made available to EMODnet Biology or your products, they need to comply to our guidance, because we don't have the resource to do so for you. That's basically the bottom line. We also will not organize or provide training specifically for your products, for your projects alone. We do organize training, which is very generally open to the community. You are more than welcome to join such training when that happens, but we will not organize a training specifically for your projects. We counted about, I think it was 40 projects, maybe a bit more, that were in various ways related with biodiversity data. We can't do 40 training sessions, which last at least for 3 days for all of you. As you can imagine, it's just too much for us. And now the good news, what we can actually do for you. So we can republish your outputs, so data products through EMODnet. That's something we can do for you. We can, as I've said before, ensure that your data are available in other initiatives like OBIS, GBIF, and the EU DTO. I've already said that. Those are existing collaborations. It's not something new for us. It's ongoing and it's continuously improved. So it's not going to be different for your project outputs. If you have products that you would submit for republication through EMODnet Biology, we need to go through an assessment and eventually choose the ones that we consider are the most relevant for our users and stakeholders and show them in the map viewer. That doesn't mean that they will not be republished. If you have a metadata record associated with your products, we can also make that visible in the EMODnet catalog. That is not a problem. It's just what I just want to make quite clear is that not all the products can be published through the EMODnet viewer. Because we are an EU service, so we have a level of commitment to ensure that these things are maintained through time. So it won't disappear after 2 years, or that's because we're not a project like the ones that you're involved in. We are something more of a long-term service. And we can, if you on the data side, if you want to publish the data via EMODnet, we can provide support with some tools and services, and so including the use of the IPT. IPT, for those of you that don't know, is a GBIF tool that is used for the sharing of data. I'm not going to spend too much time because I'm sure Ruben has that in his slides as well. And that's the end of my presentation. I hope it was clear and that it has already addressed some of your questions. But yeah, as I said, we will have some time for questions during today and tomorrow. So please feel free to ask for any clarification if you need to. Thank you, Marina. Yes, back to you.
Yes, thank you, Joanna. And again, if there are any comments or questions now, please do raise your hands. We have organized a short survey. Oh yes, a short survey to collect information of who the participants are. Unfortunately, we don't have time to introduce all of us, but we will have a short survey after the next presentation. And now I see raised hands. Yes, good morning, Niels. Please.
Thank you very much, and thank you very much for the presentation. I was wondering whether EMODnet is also bidirectional. So I'm a GBIF node manager for the Netherlands and also the European representative. So is also data from GBIF ingested in EMODnet Biology? So not the other way around only.
So
Sorry,
maybe I can say
talking.
I can answer something, Joe.
Yes, you can.
Hey,
yes.
Cheers. So indeed, in EMODnet Biology, we're gonna be gathering data that comes from GBIF that makes sense for it to be in EMODnet Biology. So as Joanna said, marine data from the European coastal waters or by European researchers, but also it works the other way around. Sometimes we also work as a data mobilizer. We have in our consortium a lot of partners all across Europe. They send and we power that data flow that also ends up in GBIF. So it works both ways. Okay, excellent. Thank you. Cheers.
Hello, Angel. Angel, please.
Hello. Good
Hello.
morning,
Good morning.
I want to know if we have in the project data sets published in public repositories, if we can provide you metadata and then link this metadata in your system to the data we have published in the public repositories.
That's not really the way we work at the moment, Angel. We do that for, we host the data. So we would need to have the data with us for that to happen. It doesn't mean that this won't change, but at the moment, that's not the way we are working. So in order to have the data available through our services, we need to host it.
So but if we have already published the data, we can provide you also this data and you can publish. If it's in
OBIS or GBIF, yes.
Yeah, okay.
Yeah. Okay. If it's in a repository like, let's say like Zenodo or like Pangaea, then it's not necessarily enough because we will need to ingest it. But if it's in GBIF or OBIS, it's already in Darwin Core. So it's much easier for us to grab those data and also have it available on EMODnet Biology.
Okay, thank you.
Yeah, without further work, that's what I mean, without any further work.
Yes, if there are no questions now, I can leave the floor to Nikola, who is going to present a synthesis of the results of the questionnaire, which was compiled by many of you. Thank you, Nikola.
Good morning to everyone. My name is Nikola. I am a colleague of Marina at the National Institute for Oceanography and Geophysics. First of all, thank you for joining us for this workshop. Today, I'd like to share with you some insights from a questionnaire that we conducted recently that was aimed to assess the best practices in data management among European Union funded projects. And this survey was aimed to understand a little bit how projects approach data collection data standardization, sharing, and some other key elements that are very important to ensure interoperability and reusability of biodiversity data. As I present these key findings, I invite you to think about how your own projects align with these practices and where EMODnet Biology could provide some support to you. So without further ado, let's start with the analysis of the results. Now we have gathered 21 answers coming from 16 different projects. The participants, the role in their projects were mostly project coordinators, researchers, or data managers. Regarding the type of data and the geographical coverage that the project data was covering, we see that mostly in most cases, the participants were gathering data from the Mediterranean Sea or the Atlantic Ocean, then the Baltic Sea, the North Sea, the Arctic Ocean, and the Black Sea. The type of data that the project was covering was mostly in situ observational data, followed by imaging and genomics data. Some were citizen science data. And the functional groups that the project data covered was benthos, zooplankton, fish, phytoplankton, and so on. We are be sharing these results with you. So if you're interested to dive a little bit deeper into this, you will have the results with you, so you can take a look by yourself. Let's dive into the data collection and storage practices. We see that mostly the size of data that the project were covering was in terabytes, and the format was in CSV format or XLX files or Excel files, followed by NetCDF files, whereas a very limited number of projects was using databases, relational databases to store data. Regarding the data management practices, in most cases, the project had indeed a data management plan. In some cases, they didn't have, which is a little bit strange, since having a data management plan is a mandatory thing for a European Union funded project. And we see that it's kind of equally whether the project were using standards or some best practices to format data or they didn't. This is a very important point that we'll dive a little bit further down this workshop. Regarding sharing accessibility, you see equally the storing of data was in cloud storage, on institutional servers, or in local storage, whereas the sharing was done mostly by data repositories and shared drives, sometimes also by emails. And now let's look a little bit more about the challenges and the gaps. We see that the lack of standardization is the primary challenge that people were facing when managing project data, followed by lack of personnel, storage limitation, lack of tools, lack of training. And the gaps that they were finding was in dealing with project data was inadequate metadata, lack of interoperability, poor data quality, insufficient training, and insufficient data backup. I've put these arrows because these are all the things that EMODnet Biology could lend a hand to you. So everything that is going to be covered in this workshop will be related also to these problems, to these challenges and gaps that you face. Now, finally, let's see the awareness and the use of EMODnet Biology data portal. Most participants were aware of the existence of EMODnet Biology. Most of them haven't used the EMODnet Biology data and products. And the most valuable features of EMODnet Biology, of the EMODnet Biology portal, was to search data and to retrieve data. Now, we have finished with analysis of the survey results, and before moving on, I will just like to invite you to participate in this brief interactive session using Mentimeter. So this will give us a chance to collect some more data on who is currently participating in the workshop. So if you have time, please join us at the menti.com website and use this code, or otherwise you can just enter the platform using the QR code. So I will give a minute for everyone just to join the platform, and then I will present the platform so we can go through the questions together. Okay, great. Let's wait. I will give you just a couple of more seconds to finish this. Okay, let's go for the next question. What is the name of the project you are representing? We should wait just for another 20 seconds, I'd say, then we can go to the next slide. Okay, let's move on. Okay, I think we can move on now. What types of marine biodiversity data does your project collect? Okay, we can move on now. What are the primary challenges your project faces in sharing data? Okay, have you used EMODnet Biology to search or download data? Okay, thank you very much. I think this was the, sorry, there was one last slide. What kind of support would you like from EMODnet Biology? Okay, we can move on, and now it's definitely the end of this presentation, and I will stop sharing my screen now. I will be sharing these results with you later on. At the end of this workshop, we will also be sharing some a survey that will assess the feedback about the workshop, but that's going to be at the end of it. I thank you very much for participating, for your attention, and well, see you soon. Thank you. I give back the floor to you, Marina.
Thank you, Nikola. Good. So this was useful to collect information from you, because we are many. Now we have time for a few questions, or we can, if there are no, we can go directly to the project pitches. So please, if you have any comments or questions so far. Otherwise, we can proceed to the project pitches. I hope I'm able to present my screen. Is it the right one? No, come io non vedo. I cannot see my screen. Where is my screen? Vieni un attimo qua. Sarà questo. Sì. Yes, can you? Yes, so I organized the pitches in this order, and if you don't mind, I will screen I will change the slides. So first we will hear about BlueRemediomics, then from Biodiversa MOSTFUN, and then from AtlantECO, BioOcean 5D and Blue Cloud, if it's okay for you. So I leave the floor to Samuel Chaffron. Samuel, are you here?
Yes, I'm here.
Okay, so
Everyone, can you
yes,
hear me?
yes, perfectly. So you can yeah, you can have your time. I
Yeah,
will
3 minutes, right?
I will
Okay.
just, yes, even a little bit more. No problem.
No, it's okay.
We have time. So I will mute myself and let you just tell me
Okay.
when I have to go on.
Yeah, thanks a lot. So yeah, I'll briefly present you BlueRemediomics. So that's a project coordinated by Rob Finn from EMBL EBI and Chris Boller from CNRS in Paris at ENS. I am myself from CNRS, researcher at CNRS, working at Nantes University and a bioinformatics team. And overall, so the aim of the project, BlueRemediomics, is basically to collect, assemble public microbiome data, mainly sequencing data, but not only, to harness new biogenics, new biomolecules, and also better study and survey ecosystem services at global scale. Now, Marina, we don't see the slides anymore. Sorry. We're seeing your Google, sorry, your Excel. I can also share if you like, huh? You're muted. Should I share?
Yeah, no, yes, please.
No, it's
Yes.
okay.
Yes, no, yes,
Yeah?
please. Yes.
Okay, no problem.
Yes.
Just a second. Can you see this?
Yes.
The same slides. So overall, there are three. So there are more than 3 work packages, but there are 3 large work packages. One which is mainly focusing on bioinformatics to collect, assemble all and annotate all this data, mainly through the Magnify database. There is a work package 2 focused on culturing, screening, culturing these phytoplankton of interest, and third work package also focus on screening novel enzymes, new chemistries in these large microbiome sequencing data. And also a fourth work package is more focused on aquaculture and ecosystem services and trying to connect omics to ecosystem services, in particular in aquaculture environments. Okay, so as I said, the most, the type of biological data collected is microbiome sequencing data, metagenomics basically, and we do this at Global Ocean. So we collect data for all publicly available data, as it is done in Magnify, which you probably know. Okay. So the data is, as I said, freely available, mainly through Magnify, ENA, of course, and it's also available through an API, JSON API. But in the project, so we will also generate new data, and in particular try to profile all species mainly at genomic level, so profiling genomes, MAGs, metagenome assembled genomes at global level, for sure presence absence of these genomes, so of the genomic biodiversity, mainly for prokaryotes, but we also include eukaryotes, and try also to profile the abundance of these at global scale. So I think this type of data would be. I think it would make sense to submit and to include these in EMODnet, probably as a netcdf and all the format you are dealing with, probably. So other users, other people can have access to through my viewers, web services as well. Okay, so that's it for me.
Thank you. Yes, thank you very much.
Okay.
Yes, I think we will have the questions afterwards. So
Afterwards,
now,
okay.
yeah, I try again to share my screen. Now, is it? Yeah, I think you. Do you see my screen? Yeah, I think so.
Yeah.
So now I let the floor to Laura Garzoli from CNR, Italy.
Hello.
Hello.
Hello, everyone. I'm here on the behalf of the MOSTFUN consortium of 9 partners. And this is a project funded under the Biodiversa Plus partnership. The main aim of our project is to try to include aquatic fungi in existing monitoring projects throughout EU. So we are talking here about microbial diversity. Aquatic fungi are pivotal in trophic networks. They are saprotroph, they are parasites, they are symbionts, they are pivotal in carbon cycling, and they are greatly understudied. So our approach is to try to fill this kind of gaps, generating new data from understudied areas, such as coastal and estuarine environments, but also glaciers. And we are collecting water sediment and leaves, but also environmental parameters. And these samples will be analyzed through amplicon and shotgun sequencing. But what we also want to do is to capitalize from existing monitoring initiatives and projects or networks. So a part of the project will be also to re-evaluate DNA samples or environmental samples or even repositories of sequences to get together data on aquatic fungi. And can you move on? And what we want to do is to complement this biodiversity data with Earth observation, environmental variables, to model species distribution, present and future, so also in light of climate change. And as for now, we are evaluating this kind of projects and initiatives at European level. So for existing samples, so if you have some kind of samples or data that you want to reanalyze for aquatic fungal diversity, now I made some advertise. I have a stakeholder questionnaire to prioritize this kind of initiative to be evaluated, and we want to give priority to initiatives that has a large global, a large geographical scale or temporal scale. And as for the interested for EMODnet, we have, of course, we are a European project, so we will make our data available open access. And our data and metadata are structured using control vocabularies such as Darwin Core. And we have our data management plan. But what I think is of interest for your initiative is that we have among partners the co-chair of EUCN specialist group on aquatic fungi that we helped to establish this group. And we also have among partners the GeoBond Secretariat, because we want to develop a new set of essential biodiversity variables dedicated to aquatic fungi. So this is to standardize the collection of this kind of information on aquatic fungal biodiversity, but also to scale up and to make our data comparable with other projects and initiatives. So here is our website, and we are evaluating the data repositories. We are in touch with the LifeWatch, but definitely I think EMODnet has a lot of potential for our data if you are interested in microbial diversity. Thank you.
Thank you very much, Laura. I think it's a very interesting approach, and I mean a different compartment that we haven't tackled so far. Thank you. Now let's go to the next presentation. So by Meike Vogt.
Yes, hello, welcome.
Hello.
I'm Meike Vogt, senior researcher at ETH Zurich in Switzerland. And I'm a member of 3 projects at present that are Horizon or Edge 2020 EU projects, AtlantECO, Bioceans 5D, and Blue Cloud 2026, that Sam and Chris and Victoria, who are present today, also are part of. So in all these projects, we're trying to understand the microbial, ocean microbial loop, or the ecosystem structure of microbial ecosystems, with the AtlantECO project mostly focusing on the link between Atlantic Ocean ecosystem structure, function, and ecosystem services. The BioOcean 5D focusing on the microbial diversity from the coastal to the open ocean and links to potential policy actions. And the Blue Cloud 2026 project being an EU infrastructure project that sort of tries to provide access to all sorts of different data platforms, to provide infrastructure, to do computation, to add value to data sets and to integrate them, and then also to produce ecosystem EOVs or EBVs associated with plankton ecosystems. The type of data we collect is relatively diverse in all these projects, starting from microscopy data, to all sorts of different metagenomics or omics-based data sets, to plankton imaging. But in BioOcean 5D, we also have acoustics. In AtlantECO, we have bioprospecting molecules and plastisphere observation, carbon fluxes, pigments, and indicators, etc. Geographical area is Atlantic Ocean to global, with a focus on the European coastal seas in BioOcean 5D. Next slide. I can't change the slide, Marina. Yes, so in all the projects, we of course have a data management plan. We're trying to follow standardized vocabularies, like in the previous project, so Darwin Core, standard notation. We are trying to figure out how to use a reasonable taxonomy with worms for anything that's eukaryotic. We following climate CFC convention for all the NetCDF outputs. And we've tried to provide project-wide data collection templates to get all the data contributors to try and produce from the start fair data that is compatible with a large range of international repositories. What we usually have as our most difficult challenge is that we have many data where we aggregate, we collect data sets, we provide some added value, so we do computation and have level 2, level 3, level 4 products, and then we're not entirely sure how to funnel those back to international repositories, and this is where we hope to collaborate with EMODnet, for instance, in terms of extrapolated maps of diversity, extrapolated habitat projections for different types of microbial, organisms or units. And yes, I think this is in one of the project. EMODnet is also a project partner and will provide data into the project. Thanks.
Thank you very much. I have just one question. So we have been in contact with AtlantECO, especially for the products, I think. Is it correct that
Yes.
some colleagues working in OGS are involved? And so I know there was an interaction.
Yes, so I think also an interaction is happening through the project coordinator, Stéphane Besan, who is leading AtlantECO, where we would hope to make all our maps publicly available through EMODnet.
Okay, yes, and Stéphane Pesant, I think he was partner of EMODnet some times ago. Can you confirm, Joana?
Yes.
Yeah.
Yes.
Yes. So he is very well aware.
If I'm not mistaken, right, Ruben? Not a partner, but
that's right.
specific data grant. Yes. Is this all the projects that we're supposed to present now, Marina?
Yes, the first session. Yes, for this. Yes.
Yes. I think we have a question. Do we? No? Well, there were
Yes. Yeah.
Yes. Okay. Sorry.
I'm told that I'm presenting or pitching from MARHAB today. Is that right, Marina?
Yes, in a later session, like at 12 o'clock, is it okay, or do you prefer now?
That is okay. That is okay.
Oh.
It's fine. Thank you.
Okay, thank you.
So we have maybe 4 minutes, I guess, before we pause for like a coffee break and just zoom out. Let's put it like that. There were a few questions on the outputs of the, and I think products as well. Yes, we do accept NetCDF files for products. Actually, that's what we are working with. Salva will explain that a bit better tomorrow, so go in a little bit into more details. I would just like to, again, encourage you that if you do have this type of a file format for the products, to check the guidance that we have, so that we don't have any issues when trying to publish the products. I mean, as probably Salva will explain, even though there is guidance for NetCDF for biodiversity products, the systems, and we have systems in place in the central portal, which will allow for the machine-to-machine communication and subsetting of the products and querying. Let's just put it in a very nice way that the implementation of those systems themselves, they do not comply with the rules that they created. So we adhere to the rules that they have defined. So for GeoServer and for ERDAP, and then we have to change things because their implementation is not following their own guidance. So we have a bit of experience in going around the constraints that we have in publishing our own products. So that's one of the reasons why we encourage you to also check our guidance, adhere to it, and if you have any questions, then come back to us. And I think that's all that we need to say at the moment. Salva, you will be following this up tomorrow, right? So we might have a few more questions then.
Yes,
the chat, Salva, is just the guidance.
Yeah, yes, I did in the chat a couple links. Yeah, but I will explain more tomorrow. So you can already have a look, but don't worry. Tomorrow I will go more in detail with this.
Yeah, Samuel. Yes.
Yes, I have a question. So I understood that EMODnet is mainly focused on EU data, right? European data.
Yes.
But do you also accept global ocean data, basically? Because in BlueRemediomics, we don't necessarily focus on European seas or ocean regions. I think that's the case also in AtlantECO, although the main ocean in the study is the Atlantic. But do you also accept global scale maps or data?
Yeah, we do.
Okay.
We do. I think this is the generic answer is yes, we do. We might need to assess what's the best way to get the data onto us, if it's directly submitted to us, or if it can go via one of the OBIS or GBIF nodes, and then we push it into, we pull it into EMODnet as well. For the products, for sure, because as you are an EU-funded project, there are no constraints on that. It's just the assessment that we need to make first, and so the minimum, let's say, support that we can give to the projects is ensure that the products are discoverable via a record in the EMODnet catalog, and that's the basic. And we can do a bit more, but that's the basic that we can do and we will do for all of you.
Okay, and as of today, do you already have global ocean maps or
we have some products from Mission Atlantic, right, Salva, that cover the northern and a bit of the southern Atlantic, which is out of scope, let's put it, for EMODnet. We are interested in the North Atlantic that is closer to Europe. But yeah, so we have a few products that cover way more than that. It's really not, we're not gonna cut your products just the European space. That's not, we're not gonna manipulate them. Yeah.
Okay, thanks.
Yeah. Marina, I think.
Yes, before we stop for, let's say, 15 minutes, can we take a picture, a screenshot of the screen? So I kindly ask if you could turn on your camera. Thank you. So it will be for the report and for the reporting. Yes, keep it open, please. Okay, one. Okay, thank you very much. And I propose it's 11:10. Let's see at what do you say, Joana? 11:30 we said.
I think we yes.
Yeah, the agenda said 11:30.
Yeah, I think.
Okay. Okay. So let's meet again at 20 minutes then. Thank you. Okay, so yes, please.
Awesome.
Yes, please.
All right, so it's 11:30, so I can start talking. I'm gonna give a presentation on the Research Data Management 101. So it's gonna be very basic. I understand that many of you are already gonna be familiar with a lot of the topics that I'm going to mention today. But I also understand that a lot of you do not have data management plans or are not familiar with the usage of data standards or control vocabularies. So I'm hoping that a lot of you also will learn something from this presentation. It's gonna last about 30-35 minutes, and I know that we're really keeping good track of the time today. I hope I don't pass too long. Because there are a lot of things to say about the basics, just the basis of research data management. So if you're familiar, feel free to just keep an ear. If you're not familiar with the topics, relax, relax and accompany me in this journey. I would like to introduce myself first. I am Ruben Perez, the data manager. I work at the Flanders Marine Institute and for EMODnet Biology, for the EMODnet Biology project here in Belgium. So the data manager definition can be very varied depending on who you ask. If you're a scientist, a data manager is probably a person that asks difficult questions about your data. But actually the definition of it comes more from I do help scientists to make their work more efficient and to deal with their data in an optimized way, so they manage everything more efficiently, so they lose less time working on the things they need to work and finding the data they need to find. And in this role here in the data center at Ville, we have a number of tasks. One of them is the very basic data management that I do for EMODnet Biology and the European node of OBIS, where I do data transformations, archiving, quality control, documentation of data, always in the field of biodiversity informatics. So we're going to be talking about occurrences biodiversity data. If you've heard, I hope so, about OBIS and GBIF, is the same scope that we have. We also work in data product, and here it can go data services, tools, applications, and for example, this map comes from a quality control tool that we've designed thanks to managing the data in a very specific way that help us to quality control not only the data that we have, but also any data sets that is in a Darwin core format, a topic that we will bring tomorrow more in detail. We also perform a lot of trainings, and here this picture is from a hackathon that we participated in as coaches, but we also do a lot of capacity development as these type of webinars or workshops, online courses, guidance, and documentation online. So there are a number of resources that we can make available for all of you in case you need it. And of course, we do all this in a holistic way We work with the academic world, but we do not stop there. As you can see, a number of initiatives here are also gathering data from citizen science projects, for example, or we're also in contact with industry or the government data, right? So it's we're trying to address a very big global problem from different angles, right? This presentation will be divided into three different sections to make it a little bit more digestible. The first part, I'll talk about why it is important to perform research data management. The second will be about fair and open data. I will explain what the fair data principles are. And in the third one, we will apply those fair data principles in the research data cycle. So we will say what things you could do in each step of the research data cycle or the life of your data to make it fair and to manage it in a good way. Let's start with the first one. Data, we need to understand data as the basis of knowledge, is the units of information. And we use this information to take decisions, right? So all the decisions we need to take are based on this basic information that is data. If we want to take good decisions, we need good data. And this is one of the first reasons why we have to manage data. And always the reason why the data is collected is to be used. And to be used, data needs to be understood. If data is not understood properly, if we don't go through the process of gathering data, extracting the information and the knowledge from it to make better decisions, we can end up in very dark places. So this is one of the reasons why, this is another of the reasons why we manage data. I have an example here of very clean, seems like a kitchen cabinet or a storage room where everything is properly stored. And this is just a metaphor for what the topic we will be talking today. In this kitchen, everything is properly organized, everything is properly labeled, and this food seems to be organized in a very easy way to find if you need. So next time you need to go shopping, it's going to be very easy to know what you already have, so you don't spend double of it in things that you already have, so you don't need. But it's also easy in case you want to mix some of these ingredients to make recipes, right? This is a way of managing this information, or in this case, the ingredients that we have in an ordered way. But this is not the only way we can organize things. We also have a chaotic environment, and do not panic in case this office resembles yours. A lot of us have been in environments like this, and here it's sometimes complicated to find the things that you have, the data, or what in case you need to perform any activities. Sometimes very hard to find the information that you need. So we will try to move away from this using a number of tools that will be the fair data principles. The last image shows what we call organized chaos. In this case, everything is in boxes. However, the boxes are not, for example, labeled. So it will be easy for you, or relatively easy for you, to find things in this room. However, when you're trying to tell others to search for something in your room, it's going to be a lot more difficult to communicate where things are. And this is why organized chaos is better than chaos, but it can still be improved. And the thing is that we are all data providers and we're all data users. So the reason for doing data management is not only to benefit other people, but also to benefit our future selves, because the first collaborators that we have are our future selves when we are looking for data that we worked in 5 or 10 years ago or 15 years ago. And we don't find it, or we do not know what the column names mean because we use abbreviations and things like that. I have compiled a couple of extracts from 2 different Nature papers. One of them from 2016 reads, students in PhD programs spend up to 80% of their time on data wrangling, fixing, formatting, and minor mistakes to make data suitable for analysis. And they say this is wasting time and talent. Indeed, 80% of the time could be invested in different things, for example, collecting more data or compiling more data, or working on different results or more results, working on the discussion, for example, and not on things that could have been solved by proper data management. The second extract, this one from 2020, talks about the reproducibility crisis and the transparency and the sometimes lack of trust that some people have in science due to cases like this one, where more than 70% of researchers that were studied here have tried and failed to reproduce another scientist's experiments, and more than half have failed to reproduce their own experiments. So this is something that can be solved easily by documenting the data that we work in. So we're making a favor to our future selves, but also sharing the data or managing the data properly. So these are a few reasons, a few more reasons why doing proper research data management can be beneficial, not only for others, but also for you. This is another metric also from Anaconda stating the same thing. People spend too much time on things that could have been solved by a proper data management plan, for example. But it also has personal benefits. It allows to work more efficiently and organized. It allows to minimize the data corruption or the data loss. And doing proper research data management helps to increase the quality of your data, either because you share it and you let other people check it, or because you check it yourself, because you've designed it in a way that is easy to develop quality checks on it, right? But it's also good for you because mainly journals these days, or many of the journals, scientific journals these days, ask for the data to be shared as well. So this is something that now needs to be done in a lot of places. But in any case, science that is increasing the transparency of science helps others to understand what we're doing, and it increases collaborations as well. There are a number of moral obligations as well. Of course, most of the data, or a lot of the data that we are been collecting comes from public resources. And since data is unique, you cannot take a measurement of temperature in 2020 of something that happens in 2016. So if you don't have that measurement, you don't have that information. So it's unique and very valuable. But it also comes at a high cost, as Zoe said at the beginning in her talk. So it's only, well, I put it here as moral obligation, but to give back to society, not only in the form of scientific papers, but also on the data that comes from those scientific papers. But in short, better data leads to better research, and it improves decision-making and the trust in science. Now, this is just a funny image on what happens when you publish your research paper, but you do not publish your data with it. So a lot of the important thing that could be reused by others to write other papers, to gather more insights, and to make better decisions cannot be done unless you publish your data. So this is another of the incentives that we have to not only publish the papers, but also the data that comes with it. And I understand that many of you come from all these projects that are asked or required or encouraged to share data. So I imagine we're all on the same boat here. But I also guess by the Mentimeter that Nikola did, that not all of you have the tools to do that. And this is why we will talk about fair and open data, and we'll see how we can make our data fair. And for those of you that are not familiar with the fair data principles, is a set of guidelines to help us to organize our data so it can be more easily found, accessed, understood, exchanged, and reused by humans, but also by machines. It's a set of guidelines. It's a list of sentences. I'm not going to go into all of them because they are already in the internet and you can read them yourselves, but I will mention some of them. So for the ones that are not familiar with them, to familiarize yourself with them. The first of all is to make your data findable. First of all, it cannot be in a locked cabinet of your desk, and people need to be able to find it. And for this, it needs to be somewhere. Somewhere can be the internet. Needs to be available online, and it needs to have rich metadata, so we can find it when we look for it. For example, using Google, something as simple as using Google. If you have rich metadata, and we will see tomorrow how in our type of data sets we can add metadata to the data sets. this is a way in which we can make our data findable. So as you can see, it's not a big deal. Another thing that we can do is to add persistent identifiers. PIDs, for example, a DOI, an identifier for each data set, so we can also trace it, we can look for it and find it. We can communicate with each other on which specific set of data we're talking about, but we can also find it in the future. We can trace it back to who it belongs to, etc. And when we talk about accessibility, we're talking about the data being retrievable. So we're not only able to find it, but also to get it and to explore the data itself. Now, accessible doesn't mean open. We've been hearing about this. Data needs to be as open as possible, but as close as necessary. And we understand that many of you will have some restrictions to share data. However, there are ways in between where we can get to an agreement, for example, moratorium periods or something like that, where data will be eventually accessible for everyone once you're done with your study, for example, but not lost. We don't want the data to be lost, right? And data can be, so we can make this accessible. We can specify authentication and authorization steps, so only people that you want can access that data. And however, the metadata should always be accessible. So even if the data itself is not, something very basic to make your data fair and very recommendable for people to find you and find your research is to put your metadata somewhere online so people can find it, ideally in an interoperable way. And this is the next letter of the FAIR data principles: interoperability. We talk about adding to our data machine readable components, so open formats, recognized standards, linked data. In short, data needs to be ready for integration. Integration is a compilation of the different data sets into one system, and for that they need to be able to speak the same language. All these data sets need to be in common formats and standards, so they can communicate with each other, just like I'm delivering this talk in English and not in another language, so we can all understand what we mean. So the idea is the same. We'll see later how it can be done. And when we talk about reusability, we're talking about data provenance. So a lot of the information and documentation needs to be added to our data, so we can know who to ask the questions about that data when we don't understand it, or if we don't understand it, find the responsible people for each step, so if there are any troubles with it, we can find it, but also so we can acknowledge those people for their work. So if you add your name to a data sets that you have, your name and role to a data sets that you've been working on, that can also serve for yourself as a personal CV, for example, not only on papers, but also on data. And this is something that I haven't mentioned before, but data papers are also an option for all those data sets that are forgotten, or data sets for which you are not aiming to publish a scientific paper afterwards, or even if you're planning it. It's a very easy way to publish data, and focuses only on the methodology and first kind of raw data, so you don't have to spend too much time in the whole scientific part of a data collection. So it can be very easily shareable, and a lot of people can implement that, can bring that into their research. So you could be contributing to science through a different channel. But of course, we need to add also for the reusability part, data usage license. We need to let people know how they can use their data. And the thing is that in many countries, data is closed by default. So if you want people to use your data, if it's not enough with publishing it somewhere, you need to also specify what the rules they can use your data with. And fair data is a spectrum. So it doesn't mean that your data either is fair or is not fair. And you can give small steps to make your data fair, either in the findability aspect or in the accessibility aspects, etc. And as I said before, it doesn't mean that, and I know that some of you have restricted data, and open data does not mean, so a fair data does not mean open data. So we can always find a middle ground and an agreement to share the data that we have, right? And this is, I'm coming to the third section, which will be a little bit longer, I hope. So I hope I'm still good in time. I think so. And we will talk about the research data cycle. I imagine you all have heard about the circle of life. And well, today we're going to talk about the circle of data, or the life of the data. And as you know, all scientific research starts with planning. Although I know that many times these research projects may start with the data collection and then later plan what they're going to do with it. But this is the most appropriate way of running a scientific project. So we have planning, collection of data, processing and analysis of data, preserving and archiving of data, sharing, reuse, and maybe after reuse, that can be the beginning of a new research project that starts again with the planning. And so we're going to break each of these steps down into how can we integrate the FAIR data principles in each of these phases. And the first one, planning. We're going to talk about DMPs, data management plans. I know that many of you are already familiar with them. So I'm just going to repeat what many of you know, but I also know that some of you don't have them yet. So I will introduce them for you, for the ones that are not familiar with them or how to make them. A data management plan is a document, is a formal document, but it's also living. This means that it can change throughout the life of the research project. And the reason is because we all know how research projects can vary through time. We all know how complicated data collection can be if we have adverse meteorological conditions, and how sometimes we need to readapt in case we don't have enough resources or enough data and to achieve our research goals, right? But in short, it is a document where it's specified what will happen, how the data will be handled during the project, during the collection of the project, but also after the research project. So we can give a second and a third and a fourth life to our data. So it doesn't stop with our research. And the reasons why having a data management plan is to save time. First of all, if we have things organized beforehand, we don't have to do it afterwards on the fly. It avoids problems, well, because everything has been thought thoroughly before the next steps happening. So you have less chances of falling into unexpected problems. It anticipates costs for the same reason. And the good thing is that it's fair by design, which means that you can design your data collection, your data analysis, your data sharing to be fair from the beginning. If you already have thought about all those things, you don't have to do them on the fly, which saves time. The content of a data management plan, it can be varied, and that's going to depend on your research. But they all have very similar sections, right? These sections are going to be a data summary and a description of your data. They're going to have metadata and documentation about the research project. You're going to have a section for data storage and security, how you're going to deal with it in the long term also, so preservation of that data, how you're going to share or reuse that data, ethical and legal aspects of it, responsibilities and costs of it. But the good thing is that you don't have to do that, to think about all that yourselves, because there are DMP templates. So there are a number of sources online that you can use. You get the template and you just apply in each section the things that, or change the things that affect your type of research project. So this is a really good thing to begin with. And the second section is about collecting data. And in collecting data, I would like to focus on metadata, control vocabularies, and standards. These touch a little bit on the findability and the interoperability aspects of FAIR. So let's delve into it. When we talk about metadata and documentation, we're talking about making things easy to digest for the people that will come afterwards. And that can be yourself 5 years from now, or it can be a collaborator or a student, for example. So we can do this at 3 different levels. The first level would be project management, for example, specifying and documenting properly the directory structure of your project. If you're going to be collecting data, you're going to be transforming that data, enriching it, and doing analysis on it. You will likely generate a lot of different files. So it's very beneficial to know how those files are going to be organized in folders. This is something very basic that we all know. But we can decide all those, for example, names of those files beforehand and the order in which the directory is structured. When we're talking about files, name conventions or basics, if you don't have one in your institution or in your project, at least you can have one that you use. And if you have one that you use, you can share that with your colleagues so they all use it. And ideally, this would also solve a number of problems that come when trying to search for data in our own directories. And README files are very famous to document data at a files level. But we also have data standards and vocabularies. Data standards and vocabularies are pretty interesting in the field of interoperability, in the aspect of interoperability. And there are many different types, but I would say the definition of a data standard can be a set of guidelines or rules that specify how data should be structured, formatted, and represented to ensure consistency, interoperability, and efficient data exchange. So it's a common framework. It's a common language to communicate with each other using machines, right? We are all familiar with some of them. For example, the International Organization for Standardization is global, multidisciplinary. We use one of the ISO formats for dates. So we always, in EMODnet Biology, we always use the ISO 8601 format. So that is year hyphen month hyphen day. But you know that there are so many other different types of formats for dates. If we all use a different one, and then you try to search information on a compiled version of all our data sets together, it's going to be a lot more difficult if we have different formats than if we have the same one. So that's the most basic idea of what a common data standard can use, no, from different formats. And we also have domain-specific data standards. This one, for example, Darwin Core, globally known worldwide in the field of biodiversity informatics. We will talk a lot more about it tomorrow when we will explain how it works in detail. But for the moment, Darwin Core can be both, right? It can be a control. It can be a list of terms. So just in fact, it is a list of terms. So a controlled vocabulary where each term has its definition, so we know what we're referring to exactly. But Darwin Core archive files, which we'll talk tomorrow, help us organize data sets and say where each field should be, how it should be called, and how the different tables of a data set talk to each other to be organized in a better way. We also have metadata standards. EML, the ecological metadata language, also very well used. Also has this is written as an XML file, and it has a number of all the information that you add in the metadata can be structured as an EML, and it can be easily shared with everyone because the wider community it's using it. And I'm talking about the wider community, and of course I always mention OBIS, GBIF, and ourselves, EMODnet Biology, because we're using the same standards. But this is already a big part of the biodiversity studying community. And when we talk about controlled vocabularies are a list of terms where each term means just one thing. So this ensures standardization. So we talk about the same thing. It's very common, for example, in my role as a data manager for EMODnet Biology, to receive a lot of data sets where they have measured biomass. However, we know that biomass, there are many different types of biomass. You have dry weight biomass, you have ash-free dry weight biomass, you have wet weight biomass. However, if they do not specify which type of biomass it is, which is very obvious for many of us, but not always for the researchers, because maybe maybe for this taxon or for this ecological functional group, it's always common to use wet weight biomass. So why do we need to specify it? It's just biomass. People know what it is. However, when you mix or merge that data with the data from all the other communities in large integrated systems like EMODnet Biology, we need to be a lot more specific when we talk about which type of parameters we've been measuring, so we do not make mistakes and try to sum, for example, parameters that do not make sense to be added to each other. And there are other control vocabularies. For example, the World Register of Marine Species is a list of names. We know how complicated taxonomy can get, from the vernacular names where people would use the same name to describe two different organisms in two different regions of the world, to the same species or the same taxon having two different names because they added those names different peoples and they never communicated with each other. We tried to solve that. The scientific community tried to solve that with the scientific nomenclature, but then we fall into the same issues because maybe one species is discovered in one place, it's published, but then another scientist can rediscover that species if it's not aware of the existence of the first one or something like that, or they don't communicate properly. It can give a second name, or we have synonyms, we have spelling variations also. So this is a way WORMS uses the power of informatics and data management to solve this problem by adding one link to each taxon and linking all the different names that exist already to that number. So it's easy to connect all of them regardless of where they are. And another one I bring to you here is marine regions. It's also a list of marine georeference place names and areas. It's hierarchical, so it can be easily used to decide which regions we are interested in. For example, when we have a large pool of data points, but we are only interested in one place, we can use marine regions to do spatial joins of that data and only focus on what we're interested in. So it's a way also to filter databases. The next part is processing and analyzing data. And here I'll talk about data curation and file organization. I have 5 more minutes if I'm not wrong. All right. In the data curation process, it goes in steps. So we always, when we collect data and we have to start working with it, we do first data exploration of what we have. Sometimes we're going to transform that data to other formats, maybe to prepare them for analysis or to share them. Sometimes we're going to enrich our data sets with information that comes from somewhere else. So I have CopyPods, but a CopyPods data set, but I want to also know what's the temperature of those places and of the places where I collected the CopyPods, and I didn't gather that data, so I can get it from another database. But again, after all these changes, we also need a validation or a quality control process that help us make sure that we have not made any mistake in any of the steps that we've made our data sets go through. For those curation steps, there are a number of procedures that can be taken care of. Most of them are very basic, and I hope you all are aware of them. But in case you are not, here I bring them. Keep your raw data intact and document all the transformations that you made. That is very easy or very straightforward when you're coding those transformation or the analysis, because in the code itself you can document each step. But I understand also that not everyone works with coding, so this is something that also needs to be done regardless of what your preferred tool is. Version control of your data, very relevant. Documenting, of course, the quality control procedures, and using open formats is just beneficial for your future self. Again, you can use whatever tools you're familiar with. You can do all these steps with all of them. And this is just an example of a data curation data set. And the one at the left side is not standardized. The one at the right side is standardized to specific formats that have been decided for that specific data set, and that make it a lot easier to filter that data. Now it's a very small data set, only 3 rows, but we're gonna end up with millions of rows in this large data system when we aggregate all the data in EMODnet Biology. So these standards will be necessary to properly understand the data that we're working with. We also have file naming conventions, very, very useful, although not that well used, I've noticed. The recommendations are always be consistent, use standards, do not use special characters or spaces, use semantic version in few counts of VO1, VO2, or subsets of that. Go from more general to more specific when you name your files. And these are two different examples of files without and with a naming convention. As you can see, the ones on the right side of the image are a lot easier to understand. And that would be all for this part. The next one is preserving and archiving data. And here we'll talk just about data archiving. I bring to you here the Marine Data Archive, or MDA, for the ones that don't know it. It's a trusted data repository for marine, coastal, and estuarine research. And the good thing that it has is that everyone can use it, and it's closed, and you can keep your datasets closed for only yourself for your personal files and projects. So you can use this one, for example. However, you can open your files to, in case you need to, you can open these files to, in case you need to share those data sets. So you have that duality. And that would be all for the preserving and archiving data. We can talk about it later. I haven't mentioned it, and I cannot see the chat from here, but if you have been having any questions through the talk, feel free to put them in the chat, and then we can talk later. And about sharing data, we can talk about searchable resources and about EMIS, which is another system we have in-house here in the Flanders Marine Institute. About searchable resources, we have repositories, for example, used for archiving and sharing data. They can be generic. If you do not know which repository you should be using, of course, you can try in these two sources, retrievedata.org and fairsharing.org. They will help you. Their catalogs are help that can help you to find the repositories that you need for your type of data. We also have catalogs. Our catalogs are designed to filter through the metadata. So if your data sets have rich metadata, you can use catalogs to find them easily, filtering by taxonomy or by date or by the name of the open data or closed data, whatever. Last, you have data portals. Data portals are often thematic, sometimes a little bit more complex than the other two because they can add interactive tools. They're looking at the data itself and to help you filter through the data and get subsets or aggregated versions of data sets. Now, this can only happen because data in these portals is already interoperable. So because they are using those standards that we've talked about before and the controlled vocabularies. So thanks to that, we can then share easily and filter easily through what we're interested in. Well, you can see here a few of these resources that you can use to find data. You'll be familiar with these. You will be working on a lot more, of course. So that would be all. And then talking about the Integrated Marine Information System, or EMIS, this is a catalog and with metadata about all data sets that are marine or coastal, right? Open and not open. It's linked to the MDA. So, and we will share later a screenshot of how a record looks like. But a good thing about it is that of course it has a DOI for each data set. It contains one record per data set. And in that record, all the information, it's already connected through links within EMIS. So, for example, we can go to any of your institutes. We can click in there and find a person, everyone that works there. If you want to search what project this person is working on, you can click on the project. So everything is linked, and you can go clicking through everything to find that information. So the next step is reusing the data, and this would be the last of my presentation. I'm going to pass my talk a few minutes, so apologies about that. But about reusing data, we will be talking about provenance, documentation, and licenses. So regarding provenance and documentation, we said it before, it's about having everything needs to be properly documented so people can find it, so we can acknowledge people, and so we can find who we need to contact to talk about this data in case we have some questions or doubts. So it's just healthy for all the data sets to be documenting all the information through data lineage, for example, everything that has happened to that data set. The same thing about the license and credits. We have the Creative Commons licenses. You can have your data being open there. You can assign, in case you don't want the data to be shared, to be used with commercial purposes, or you don't want derivatives of your data because you trust whatever changes they can do to them, you can add those licenses. Of course, we recommend open licenses like CC0 and CC BY, but we can indeed discuss a little bit more depending on your data sets. I think the last two screens slides I have are about metadata and data and the relation. Data is the thing we need, right? But metadata is what we need to find the data, to use the data, to reuse the data, or to access the data. Sometimes our metadata is going to be the key to the data, and we can only access it thanks to having the metadata. That's why it is important. Sometimes metadata is going to help us to find the data, right? And to discard everything that we don't need, right? Because this world of over documentation, and we need to be able to decode what we're interested in and what we're not. And these are just a few more examples of it, and they provide context. And this is my last slide. This is just the EMIS record, so a data set, the record that we have in the EMIS data catalog. You can see here it has a title, it has a citation, a few clickable buttons to download the data in different repositories, a DOI, previous versions of these data sets, which help a lot to point which data we've been working on specifically, contact details, license, and all. The contributors, of course, are below there in that page, but I didn't show it here. And I think I've passed my presentation a few minutes, so apologies about that. If you have any questions, either we can talk in the Q&A, or you can text me here. Thanks a lot.
Thank you very much, Ruben. I propose that if there are any questions, as your presentation provided a lot of information, to handle those questions now. I just saw a raised hand. Somebody raised his hand. No. And I confirm that all the presentations will be provided by you together with also the recording.
Thanks a lot, Luke. I'm happy you like the presentation. Cheers.
Yes. So tomorrow you will be again providing more detailed, more in-depth information on how to handle the data. So if there are no comments, no questions, if not, then we.
So sorry, do you hear me?
Yes.
I left a question in the chat, so perhaps you can have a look there. I just wanted to know if one create new data, for example, shapefiles or CSV tables, are there existing tools for R, Python, or online to create or modify the metadata?
Yes, and tomorrow we will be talking a lot more about that in detail for biodiversity occurrences data. We will share a number of tools that you have to document the metadata. For example, in this case, we will be using the IPT, or Integrated Publishing Toolkit, which will guide us through all the metadata that needs to be added to a data set, to one of these data sets. And I will see, because you see these presentations usually are in the form of workshops that last 2 or 3 days. So this is a very short delivery of everything that needs to be known about this topic. Tomorrow I'll try to, let's see if we have time to do a demo of how the IPT works. A very interesting tool to share data that deals with the mapping to Darwin Core standard, but also the metadata to the EML standard. So really, really good. So there are a few tools. Let's see if tomorrow we can delve into them a little bit more.
Okay, great.
Okay, so I don't see any hands. So now let's go back to the projects. So I will start with the new projects, the new session on contrast, be useful, and MARHAB. I will come si fa? Nikola, vieni un attimo. Oh yeah. No? Perché non lo vedo? Non vedo più la mia presentazione. Sorry. Allora, questa, no? Ah, ok. No. Just one moment, sorry. Qua, ok. Yes. Can you see the presentation now?
Yes, Marina, we can, yes.
Yeah. I cannot, but okay. So I will, yeah, this is the one. Yes. So I leave the floor to my colleague, Stephen from the NIVA institution. Yes, please, Steve.
Yes, thank you, Marina.
Hello.
Hi
Nice
there.
to see you again.
Yeah, so I'm Stephen Brooks. I work at the Norwegian Institute of Water Research in Norway, NIVA, and I'm coordinator of the project Contrast, which is looking at the impacts of chemicals of emerging concern on the marine environment. So it's been started in January 2024, and it will run till, it's a 4-year project, so it'll finish in December 2027. And as I say, we're looking at the chemicals of emerging concern. So one of the big parts of the project is to sort of evaluate which of the chemicals of emerging concern we want to work in within the project. And so there's a prioritization scheme within one of the work packages. And then once we know which chemicals we're going to look at, we're going to investigate those chemicals and the impacts on the marine environment. Certainly look at biological effects data of target organisms such as fish and mussels and different species. And also a big part of the project is looking at the biodiversity in sediments and water and how CECs impact that biodiversity. In terms of geographical areas, we're mostly focused within the European seas. So, and we have some case studies which are in the North Sea, Northeast Atlantic, and the Mediterranean. So those are our target locations. Maybe next slide, Marina. And so in terms of data, we have a data management plan in place. We have OGS and Marina in charge of this aspect as well of the work. So we know it's being carried out to the FAIR principles and using standard templates. And she's guiding us into how we're going to formulate the data into final insertion into EMODnet. The types of data we collect are quite varied. So we have chemical and physical chemical data, looking at the CECs, particularly in different matrices, water, sediments, and biota, different oceanographic parameters. But then we have biological data, which is more relevant for this workshop, and where we're looking at biological effects assessments, so biomarkers in fish and in mussels, for example. But also a big part of it is looking at biodiversity, where we'll be using sort of novel techniques and sort of standard techniques in order to look at this. So some visual inspection, but also eDNA methods, proteomics, and sort of anchoring that in visual IDs, and also robotics or semi-automated systems of identification. So that's going to be done in sediment and in water systems as well. We're also creating all the data on computational data on models, and look, this is mostly looking at the sort of transport of chemicals in the oceans. So yeah, that's all I had to say, really. Thank you.
Yes, thank you, Stephen. And I confirm that this project collects very challenging data to be collected, because biological effects so far have not been tackled by either EMODnet chemistry or by EMODnet biology. So we have to figure out how to handle them. Thank you.
Thank you.
So the next project is Be Useful. Very nice acronym. And so I'll let Julia speak.
Yeah, thank you, Marina. Okay, first of all, thank you all for these presentations. I'm here mostly because of my interest in what EMODnet is doing and the possibilities that we have through EMODnet, and also learning from you all and your data management plans, which is pretty new to me. So I'm probably not the best person to share about data management plan of my project, which is be useful. But luckily, there's more people in this meeting that are from my SEM project. I'm more used to working with the MPs. So welcome to ask, and I welcome them to answer for me. So Be Useful is this European project that is coordinated from the Technical University of Denmark by Martin Lindegren. And well, I'm Julia Polo. I'm a postdoctoral researcher at University of Tromsø in northern Norway. And the aim of the project is to basically operationalize biodiversity and ecosystem services indicators through end users co-developed decision support tool. The project is very focused on the co-development of these decision support tools and the sharing species distribution maps and information on biodiversity and ecosystem service to make it actually useful to conservation and management, which I guess is usually very repeated wording across European projects, but we try to make it the main focus. So we are not collecting new data. This is important. We are using and accessing and reusing already published data from regional seas all across Europe, Mediterranean, North Sea, etc. For benthic, demersal, and pelagic fish and invertebrate communities data. For those communities, we are computing biodiversity indicators, which we hope to be able to model through HMSC, which are hierarchical species distribution models. And those would be converted into outputs, which are the main focus of the project, which we expect to share. So the idea is for European regional seas to identify areas of particular interest in terms of biodiversity and ecosystem services and provide useful tools for conservation and management. Next slide, please. So this is just an example of what we are doing. I put this image here just because I wanted to. I think it's a very graphical way to say that we will be using and we are using loads of data from very different origins. Lots coming also from EMODnet service, data on human activities, on abiotic and biotic drivers and pressures for the marine ecosystem, and also for biodiversity. So as for our data management plan, for now, the final data management plan would ensure that all outputs are findable and accessible. And the idea is that they are published in online catalogs like ICES, Geo Network, following the INSPIRE guidelines. And I think it might be a possibility that EMODnet platform is also accessed through the project. So this is my pitch. Thanks a lot.
Thank you a lot, Julia. Thank you. And now the next and last for today, for this morning, yes, is the project MARHAB. Yes.
Thank you so much, Marina, and it's great to be with you today. I'm presenting this project that we have called MARHAB. And the full name of the project is Improving Marine Habitat Status by Considering Ecosystem Dynamics. And I will get a little bit more into detail in the next slide of what that project is doing. My institution, Institute of Marine Research. No, so you have to go back. Sorry. I'll tell you when to switch. So I'm the coordinator of the project on behalf of my Institute of Marine Research, Norway. And we are collecting data on coastal fish assemblages by means of BRUVs, so stereo video images, marine animal movement in terms of both acoustic telemetry and satellite telemetry, genetics, genomics, and we will also quantify function. So we will look at predator presence and size structure. The geographical area or the case, we have case studies in Skagerrak and Kattegat, but our project has a wider geographic scope also. And I'm happy to see that two of the partners, Barbara Horta Costa and Ines Sousa, are here from CCMR University of Algarve. Something funky happened with my logos, but that's okay. And there's QR code that you can scan, also appearing in the next slide, I think. So if you change slide, thank you. So this is the idea behind MARHAB, is that we have taken the science on the left side of the slide at face value. We have accepted the top-down trophic interactions in marine ecosystems, and we proposed a project where we will look at how this is actually possible, both in conservation and management settings. And we will look to what degree this is achieved in present management, and we will give advice on future management. And if you follow those two half circles, you end up with the bottom box there saying that we will reconcile marine conservation with fisheries management, meaning that some of the attributes that we are looking for when we protect marine ecosystems, we think that those are achievable also in fished settings. So for instance, introducing maximum legal sizes for important top predators that provide ecosystem functions, etc. So that this is the aims of MARHAB summed up in one slide. Next slide, please. So we do have we have developed and we have an accepted data management plan, and after learning about EMODnet and seeing that our data seem to be so very much within the scope of the data that EMODnet Biology wants to collect and make available, I will certainly make sure that we strive to do that. I know that we are sharing the tracking data with the European Tracking Network. I don't know how that is interoperable with EMODnet. We are in our first year. It's a 4-year project, so there is still a lot of time to adjust our routines and to inform the way we conduct the project. And please follow us on LinkedIn and also on the MARHAB project page, which is still under being developed and scope expanded. Thank you very much for the opportunity to present.
No, thank you very much, Even. Yeah, I see Angel, please.
Yes,
Go ahead.
yes, thank you. I have a question for Julia, and be useful because you have not commented on the length of the project, on when it started and when it's going to finish, and especially when the products you have commented are going to be available.
Oh, sorry. You can hear me, right?
Yes.
Okay, thank you. I'd say I've been incorporated not so long ago to the project, but I'd say it's until 2027. Is it that way? Walter, can you confirm that?
I think 2022, 2027.
Yeah.
Yeah.
Thanks a lot.
And there was the second question of Angel was regarding products, I think.
Yes, when will be available?
From which of the projects?
from
for the
reuse
useful, right?
reuse
Yeah,
for
yeah,
this.
yeah. Can you answer that, Walter?
Yes. Since I think that the HMSC mapping results will be mostly available in 1 year since now. Since we are still working on the production of these and the finalization of these models at both Mediterranean and North Sea level. And so I think that mainly at least 1 year till now.
Then, Johanna, please go ahead.
Yes, just 2 comments very quickly. One, following Julia's presentation, if you're not collecting data and reusing existing data, and this applies to all of you, we need to be careful so as to not create duplicates if they are already published in EMODnet Biology, in OBIS, GBIF, whatever. So the process for those projects that are using data that already exists, especially data that are already published, is not exactly the same as for those that are collecting data, because we don't want to create duplicates in the system. So we would need to do a more in-depth assessment before we just ingest the data and push it out through EMODnet Biology. Might not be the case for all of you, but just that side note. And just for Evan, I imagine that Ruben will mention maybe something about ETN, but we do have a data flow established with ETN, so that should not be problematic. And I would just leave it like that because I don't want to, yeah, cool. I don't want to steal more of Ville's time. Sorry, Ville.
Yes. If there are no other questions, then it's time for EMODnet Seabed habitats. Yes. Please, Ville.
Hello. Yes, can you hear me?
Yes.
Great. Okay, I will try and share my presentation through this. Okay, can you see it?
Yes.
Great. So, good afternoon, everyone. My name is Ville. I am the coordinator of the EMODnet Seabed Habitats. I work at the Finnish Environment Institute, Syke, in Helsinki, Finland. I will be giving you a short overview of what is EMODnet Seabed Habitats, what kind of products we have, and what kind of workflow we have for publishing the data, and maybe some recommendations too. We are a consortium of 15 partners and subcontractors, and like other EMODnet thematic lots, the Seabed Habitats runs as a 2-year MFAF project, which is tendered for each 2 years, and then a possible continuation of 2 years might be offered. Our current phase is running until September 25, so next year, and we expect that it will be continued until 27 for another 2 years after that. And our products are available, some of them at the EMODnet Map Viewer. That is more of a showcase of few products. And the majority, the rest of them, more than 1,500 different layers, are available at the EMODnet products catalog and through different services like web map services and web feature services. So you can put them in your own GIS software too. Our products are usually grouped into 3 different categories. We have the modeled seabed substrate, the seabed habitat map for European waters, the EU CMAP, and then 2 other regional versions of that, one for the Caribbean, and in the last phase, one was produced for the Caspian Sea. In this current phase, our geographical scope includes all European seas and Caribbean waters, focusing on the overseas territories areas in there, but also other Caribbean areas. Other products, the majority of the things that get published are different types of habitat survey data, habitat maps, and models. And then as a third one, we have the so-called composite products, which are products that we produce ourselves from these data. The first of these, the EU CMAP, it's a modeled broad-scale habitat map for all European seas. It incorporates different kinds of background information like the bathymetry from the EMODnet bathymetry digital terrain model, biological zones layer created by EMODnet seabed habitats, which is modeled using continuous environmental layers and in situ biological data, and substrate type layer created by EMODnet geology, which is it's a compilation of substrate maps gathered by National Geological Surveys with some additional data from seabed habitats partners. And the modeling process for EU CMAP produces a seabed habitat map, which is then published in several different classification schemes. The Marine Strategy Framework Directives, benthic broad habitat types, and is the first one. This one can be used, for example, as a baseline for MSFD reporting by the member states. Other classifications that it's published in include different UNIS versions, the older one from 2007 and a new one, and also some regional ones, such as for the Baltic Sea, the HELCOM hub, and for the Mediterranean, the Barcelona Conventions classification. All these and all the environmental layers used in the modeling process are also made available in the catalog. Then the survey data and habitat maps category, it includes, for example, point habitat data, different kinds of habitat maps from all around Europe and Caribbean too, and modeled habitat maps as well. And these have both modeled habitat maps and habitat forming species models too. For example, in this photo or in this picture, the Focus model from the Baltic Sea in Finland. Then the composite products, the third category, we have, for example, the essential ocean variables for Europe. It's a compilation of different data sets, and other ones include the macroalgal canopy cover, mangrove cover for the Caribbean, and live hard coral cover in Europe. And besides these, there are the essential fish habitat maps, coastal wetlands dataset, and as a new product, we are preparing a vulnerable marine ecosystems product in this phase also. And some of our work also includes this kind of special tasks which are to benefit the regional sea conventions. For example, this OSPAR threatened and or declining habitats layer that was used in their assessment by their and made to them by their specifications. The data ingestion process by EMODnet seabed habitats works so that for each of these 2-year project tenders that specify the tasks, the consortium prepares a list of known habitat datasets to be acquired. And besides these, during the project phase, new ones are identified and acquired as well. And all the gathered datasets are formatted using our own guidelines and harmonized to follow INSPIRE specifications, and some other checks, such as geometry-related ones, are done and all the metadata records are also harmonized. And then the data is published. We have a twice per year schedule to do that. And something about the general recommendations for projects. I think you might be familiar with this one. It's guidelines by EMODnet data ingestion published on the EMODnet website. Quite a general kind of guidelines which emphasizes data management plans and fair data principles, as we've heard today, and also recommends engaging EMODnet early in the project, because especially data ingestion thematic lot can assist in these planning tasks. But I'd also like to say that if you have any specific habitat data publishing related questions, please feel free to contact us at Seabed Habitats. And for example, if you already know when your project ends, if that falls into the next few years before end of September 27, then now would be a good time to contact us so that we can plan this together for our next phase, that we can adjust our own work time and tasks to assist you in the work. And yes, then for the last slide, I have some sort of a case study, because I happen to also work with the Obama Next Horizon project, observing and mapping marine ecosystems, next generation tools. So in this project, we have 12 different learning sites all around Europe, and there is a lot of different activities going on in these learning sites. For example, this one from the Baltic Sea, from the Gulf of Finland, there's both there's fish stuff going on and pelagic work and benthic studies and so on. So it's a bit of a challenge to how to effectively gather all this data, how to standardize it, do quality control and publish all this. So we have in the project a data management plan, but it's still quite general in the way that I think the next step will be that together with the data manager and the learning site coordinators, we are going to specify the data types and the timetable that they are going to be produced in, and then engage with EMODnet data ingestion and all the relevant thematic lots such as biology and seabed habitats to begin this work of ingesting the data into EMODnet. That's all I have. Just if you have any questions on seabed habitats related things, please don't hesitate to contact us. Thank you.
Thank you, Ville. It was very interesting also because it's complementary with EMODnet, with some products that we develop. So we are perfectly on time. We have time for some questions, if there are any, because it has been a long morning full of information. As anticipated before, both the recording and the presentation will be available after the workshop, together with the analysis of the surveys, of the feedback that you provided with the Mentimeter before. So, Joana, do you want to conclude the session of this morning?
Yes, I mean, I think it went quite well. I hope that this first morning has already given you information that you didn't have. We will be having a bit more in-depth information on data and products guidance tomorrow, with various links, and I think a bit more detailed on the processes, some tools and how to use them, and what we do in EMODnet Biology for the data and the products to be published. Yeah, I think, thank you very much. I think it was quite good. And please feel free to ask any questions either via the chat or yeah, just raise your hand. The idea was to make this as interactive as we can because it is an online workshop. So we don't want you to get bored and leave this workshop with the feeling that it was a bit of a waste of time. So please do ask questions that you might have today or tomorrow. That's what we're here for. Thanks, Marina.
Yes, I just put the agenda of tomorrow, so you just to remind you. And yes, I think we can conclude for today. So I really thank all the participants and all the speakers, and see you tomorrow then.
Bye. All right.
Okay.
Thank you.
Okay.
Thank
you.
Thank you. Goodbye.
Bye.
Bye. Thank you.
Bye.
Thank you to
Bye.
all. Registrazione.